Welcome to Roadmap to Residency Evidence-Based Application. And in this video, we will be talking about viral structure and replication of the virus. So virus structure, they contain nucleic acids, which can either be DNA or RNA. These nucleic acids are surrounded by a protein coat, which is called as capsid. And sometimes these viruses can be uh, uh, enveloped, which is called as an envelope, but sometimes it can be naked, and those viruses are called as naked or non-enveloped viruses. And these and the viruses do not have their own metabolic activity. They rely on the host for their metabolic activity. So capsids. So most capsids have one of the two common shapes. They can be either icosahedral or they can be either helical in shape. Whereas talking about envelopes, so viruses are naked or enveloped. Envelope is a lipid membrane which is acquired from the host cell during assembly of the virus. So they usually from the host cell plasma membrane and sometimes with the host cell's nuclear membrane or endoplasmic reticulum. Viral glycoproteins are often embedded in the membranes and they are used for binding to host cells and also antigens for immune system. All naked viruses have icosahedral capsids and the enveloped have icosahedral or helical uh, shapes. So enveloped viruses are hepatitis B viruses. Some of the examples include hepatitis B viruses, herpes virus, HIV virus, and some of the examples of non-enveloped viruses are adenovirus, rotavirus, rhinovirus, hepatitis A virus, or hepatitis E virus. So RNA virus genome. So most are single-stranded and linear. Exception is the reovirus, which has double-stranded RNA genome. Also, retroviruses, which have single strands, but they are two in number. Circular viruses, for example, Bunya virus, Arena virus, and Delta virus. So we can remember by the mnemonic BAT. So Bunya virus, Arena virus, and Delta virus, they have a circular genome, whereas most replicate in the cytoplasm, but influenza and retrovirus replicate in the nucleus. So RNA virus polarity. So the RNA virus can be either positive sense RNA virus or negative sense RNA virus. Now this positive or negative represents the polarity of the virus. So the positive stranded RNA, which is totally similar to mRNA in cytoplasm, is used for protein synthesis immediately. Then these are the positive sense viruses. And if the virus has a negative stranded RNA, which must be converted to the positive RNA first, and then can be used as a template for protein. So virus must carry enzyme to convert negative to positive RNA. So these are the negative sense virus. Okay, so negative stranded RNA, negative sense virus, and positive stranded RNA, positive sense virus. But remember that all the negative stranded RNA viruses must have an enzyme to convert the negative stranded RNA to positive RNA first for the function. RNA polymerase. So human cells make RNA from DNA through the process of transcription, and the enzyme that we use as a human is DNA dependent RNA polymerase. But viruses must make RNA from RNA. So they must synthesize their own enzyme, and that enzyme is called as RNA dependent RNA polymerase. Similar. Now, this is basic of RNA virus replication. So the positive RNA, they can act on the ribosomes or which is present in the cytoplasm to form proteins, or through the RNA polymerase, they can form either negative RNA. So this negative RNA can again be acted upon by RNA polymerase, and these can go on to form viral genome. Also, we have seen earlier that for the negative RNA to form a viral genome or to do any other function, these must be converted to positive RNA. So here it is what it is. So negative RNA acted upon by RNA-dependent RNA polymerase to form a positive stranded RNA, and then again it can go on to form protein or viral genome. So retroviruses. What happens in the case of retrovirus? So the positive RNA, they are acted upon by reverse transcriptase, and this reverse transcriptase forms the double-stranded DNA. These double-stranded DNA can go on through a function or a process of transcription to form messenger RNA. These messenger RNA can then form the proteins. DNA virus genome. They can be either circular or linear DNA. Most have double-stranded DNA, but in case of parvovirus, there is only a single-stranded DNA. So parvovirus is one exception. So viral genome infectivity. So the positive RNA genomes, they cause infection by themselves. The genetic material begins producing new proteins or nucleic acids on entry into the cell ASAP. And for example, double-stranded DNA genomes also they cause infection by themselves. But in case of negative RNA, they are not infectious by themselves, and they require RNA-dependent RNA polymerase to reproduce, and then they cause when they get converted into positive RNA, then they cause infection. Where we will also talk about segmented genomes. So many RNA viruses are segmented. Multiple molecules of RNA are present in the virus. It allows for the reassortment of RNA. So, for example, two viruses can co-infect the same cell, and the mixing of segments can form a very new virus. So, most important example for that is the influenza virus, and the other examples are Bunya virus, orthomyxovirus, which can cause influenza, arenavirus, and reovirus. Now, this is a very dangerous mechanism for human being. So DNA virus. So DNA viruses can be either naked or enveloped. The examples of naked viruses are parvovirus, adenovirus, papilloma, and polyoma virus. And the examples of enveloped viruses include hepadnavirus, herpes virus family, and pox virus. All DNA viruses have icosahedral capsid except pox virus, and all DNA viruses replicate in nucleus except the pox virus. Now here we have the classification of RNA viruses. So RNA viruses, there can be either icosahedral capsid or helical capsid. So icosahedral, there can be again naked or enveloped. So naked viruses include calcivirus, herpesvirus, reovirus, picornavirus. Enveloped viruses include coronavirus, retrovirus, flavivirus, and togavirus. And helical envelope include Bunya virus, orthomyxovirus, arenavirus, delta virus, paramyxovirus, rhabdovirus, or filovirus. You need to remember these names. All single-stranded except reovirus, which is double-stranded. All replicate in the cytoplasm except influenza and retroviruses. All positive are icosahedral except coronaviruses, and all linear except the BAED that we have seen earlier in the slide, the Bunya virus and all are non-segmented except the Boer. Now, this is a viral life cycle. So first of all, there is attachment. So for example, the influenza virus becomes attached to a target epithelial cell. Then there is the penetration. The cell engulfs the virus and by endocytosis. And then there is uncoating. The viral contents are released inside the cell. And then the viral RNA enters the nucleus where it is replicated by the enzyme viral RNA polymerase. RNA-dependent RNA polymerase that we have talked about. And then there is assembly of the virus where the new phase particles of the virus are assembled. Then the new viral particles are made and released into the extracellular fluid, and the cell which is not killed in the process continues to make new virus. Now, there is very important concept of lytic and lysogenic cycle of the virus. So first, let's talk about lytic cycle. So the phage attaches to the host cell and injects the DNA, okay? And inside the bacterial or host chromosome. And then the phage DNA circularizes and enters the lytic cycle, or it can enter the lysogenic cycle. In case of lytic cycle, the new phase DNA and proteins are synthesized and assembled into the virions, which are the multiplied copies of the virus, and then the cell ruptures. And when the cell ruptures, there is release of these sister or sorry, the release of these daughter virus or virions. Then again, it can go on to act on other cells. Whereas after the phase DNA circularizes and enters the lytic cycle or lysogenic cycle, in the lysogenic cycle, the phase DNA integrates within the bacterial chromosome by recombination. So it can infect the bacterial chromosome by becoming a prophase. Then when these cells divide, and when these cells divide, the lysogenic bacterium reproduces normally because there is integration of these DNA of the virus, and these cells divide, and there are many cells division that is occurring. Occasionally, the prophase may also excise from the bacterial chromosome by another recombinant event, initiating a lytic cycle, which means that either the prophase, or you can see the viral DNA gets attached to the chromosome of the bacteria, and the bacteria divides, and then it divides with the bacteria, or it can again go on to a recombination phase where again the viral DNA, these two, sorry, when again these two viral genetic component, these two can again get attached, and again these only genetic component can go on to enter the lytic cycle. Thank you for watching. Keep studying hard.